So it's my real pleasure to introduce Dr. Craig Thompson. And before I do that, I'll just remind you to log into the Connect Science website where you could see the schedule of upcoming talks. And also please submit your questions via the Q&A tab at the bottom. So Dr. Thompson is originally from Cambridge, Mass. He studied medicine at University of Pennsylvania and then came to Boston to do his internal medicine training. He spent time both at the Brigham and at University Hospital Boston, now Boston Medical Center. He then went to the Naval Medical Research Institute, where he spent a few years studying blood cell activation before completing his fellowship in medical oncology at the Hutch in Seattle. His independent career took him to Michigan, then to Chicago, and then to Penn, where he was a cancer center director for several years, and now to Memorial Sloan Kettering, where he's president and CEO. It's really difficult to adequately summarize his accomplishments without taking up the whole hour. But the very brief version is, his early studies were seminal in investigating the role of MYC in oncogenesis. He also had a number of seminal papers on the role of apoptosis repression by BCL2 family members, including the cloning of BCL-XL and many other studies. And more recently, he's turned his attention on the role of metabolism in growth control. He's received numerous awards, and these are equally difficult to summarize, but from institutes such as the Howard Hughes Medical Institute. He's a member of both the National Academy of Science and of Medicine, and really many others. And in my opinion, what's really most impressive about Dr. Thompson are the accomplishments of his trainees, who are really now some of the most innovative leaders in the field. So without further ado, Dr. Thompson.
Sorry for a few of the technical details. I'm really thrilled with Alejandro and Dr. Poliak inviting me to participate in this series that you guys started out at the start of the pandemic, and is an emblem for all of us of how we can continue our scientific communication as we go forward. I really appreciate the introduction, and I would really pleased to share some of the results that have just been published. So if I don't do a good job of presenting it, some of our latest insights in how cells sense amino acid availability to control transcription and translation have just been published in eLife. So with that as an introduction, I'm really pleased to tell you that story, starting with when we first became interested in amino acids, which was now almost 20 years ago. The person who has carried out all the new work that I'm going to show you is Natasha Pavlova. I'm incredibly proud of a connection to Boston, since Natasha was in fact started out in Boston getting her PhD at Harvard Medical School. And then we were lucky enough to get her to be interested in metabolism to come and join us. A number of people in the lab on the left side have helped her, starting with William Palm, who got us interested in some of the issues that Natalia has solved, as well as their collaborations with the Tuvison and Rabinowitz lab, as well as the lab of Daphne Barsaga, who's been our long-term partner in much of what I'm going to tell you about. I got to start with disclosures. So here's my disclosure slide, but everything that I'm going to talk about today has been done as part of my job in running a laboratory at Memorial Sloan Kettering. And our funding for this work has been from the NIH and from the Damon Runyon Cancer Foundation. I'm going to lead with the punchline. And the punchline is that, in fact, we have been asking for a very long time how cells get the building blocks for growth. The vast majority of the mass, or not the vast, but the majority of the mass of a growing cell is actually the proteins that make up a cell. Lipids and amino and nucleotides are the second most common. They only make up about 20% each, whereas the major mass, 60% of a cell, are the proteins that are the constituents that allow a cell to function. So how cells take up amino acids is a critical decision. And one of the things that we discovered that started this project back in 2000 was the realization that normal, non-transformed mammalian cells lack the cell autonomous ability to take up amino acids that are normally supplied in the extracellular environment because we eat. The liver normalizes those amino acids, and they're present in the circulation in the extracellular environment. The ability of cells to take up amino acids in bulk is entirely controlled by growth factor signaling. And our first discoveries in this field were to link receptor tyrosine kinases that were growth factor receptors to the signaling that allowed cells to put amino acid transporters on their cell surface and take up the free amino acids that exist and were supplied constantly by our circulatory system. We identified initially that control of signal transduction initiated with receptor-associated tyrosine kinases, leading to the activation of PI3 kinase, and that the key effector molecule for placing nutrient transporter on the surface was AKT. That AKT's role at the membrane was to phosphorylate transport proteins that regulated cycling of amino transporters from an endosomal storage pool to the cell surface. And as long as AKT was constitutively active, cells stayed and had increased dwell time, those transporters on the cell surface, and cells were facilitated to take up the amino acids down their concentrations into the environment, and that fueled growth. And I'm going to show you the data about that in a minute. A few years ago, in trying to understand the more ancient pathway by which eukaryotic cells take up, which is to take up their environment in bulk by a process that is known in slime molds as macropinocytosis, we discovered an alternative source of amino acids was to take up extracellular proteins by creating a macropinosome in which an evagination of the cell surface is initiated that circles back on itself to encapsulate the fluid phase environment around the cell, intercalate that into an endosome, and then transport that endosome to the lysosome, where their pH-dependent proteases and lysases would degrade it, and the amino acids would be assimilated from the lysosome into the cytosol to supply amino acids for growth and protein biosynthesis. That pathway also, we discovered in the last 3 years, is regulated by receptor tyrosine kinases and requires the activation of PI3 kinase to initiate a series of events that occur at the cell surface coordinated by PI3 kinases to form the extracellular evagination and initiate macropinosome formation and endocytosis. Although PI3 kinase is essential for the initiation, the gain knob on this pathway is regulated, as we and Daphne Barasaga were able to identify a number of years ago, is controlled by RAS. And so RAS increases the rate at which this process of taking up nutrients goes on in a cell. And it provides a backup system, we believe, for the take up of nutrients when there aren't sufficient nutrients in the extracellular environment or amino acids in the extracellular environment to fully supply a cell's translational needs. But as I said, the punchline of this is all amino acid uptake that we have been able to identify to date is controlled by growth factor signaling, and all cells depend on the paracrine signals in their normal environment in tissues for the ongoing needs of taking up amino acids through the regulation of these two pathways. And so if you think about it, what this really says in this first slide is that as we became metazoan organisms, receptor tyrosine kinases evolved to take the job of allowing the body, the rest of the cells in our multicellular body to control the uptake of individual cells of the nutrients that we share. And so in that way, it drives the need to match the needs of the cell with the needs of the organism. So we've gone on and explored this, first looking at what controls the uptake of amino acids downstream of PI3 kinase and AKT. I'm going to show you briefly a model. We tend to use hematopoietic stem cells because my lab functions in immunology and hematopoiesis when we look at normal cellular processes. So these are non-transformed hematopoietic stem cells that are being maintained in culture with IL-3. They're mouse cells in this case, but we've done the same equivalence with human cells. And in the presence of IL-3, what we're imaging on here is the surface expression of the backbone of all the bulk amino acid transporters. That gene was originally, when we cloned it, called 4F2, but is now known as CD98. And it's the backbone of all the major bulk amino acid transporters. And you can see in the presence of IL-3, illuminated in the upper left-hand panel, that in fact, when IL-3 is present and cells are exponentially growing, as they are in this culture, all of the transporters on the cell surface, and they're mediating uptake of bulk amino acids that are present in the tissue culture fluid. If we take away interleukin-3, just interleukin-3 from this normal cell population that's proliferating in culture, in 12 hours, all of those amino acid transporters are processed off the surface, are directed to the lysosome, and the image on the upper right-hand side in the absence of IL-3 is recognizing their ongoing degradation in lysosomes. And 12 hours later, there will be nothing visualized by this antibody. All of the transporters are gone, and cell loses the constitutive ability to initiate amino acid uptake, even though the amino acids are present in the extracellular environment. And this cell starts a clock that lasts for 36 hours to commit suicide by apoptosis. And that's how we discovered this pathway in the first place. If we constitutively activate AKT, which I said was the effector of this, what you can see in the presence of IL-3 is still all of the transporters are on the cell surface. But now if we take away IL-3, they're all still on the surface. And now the cell is cell autonomous and remains able to take up amino acids and fuel protein translation. And so this pathway is the critical pathway that controls amino acid transporter uptake. We discovered in 2003 by studying what's needed to keep the transporter on the cell surface from a genetic and a signaling pathway that one key effector that's required downstream of AKT to keep the transporters on the surface is the molecule mTOR, and specifically the mTORC1 complex of mTOR. And so that's a demonstration here. The ability of AKT transformed cells to maintain the transporter on their cell surface in the absence of IL-3, which is what's being imaged here in the upper program, when TOR is allowed to be on, is they're all on the cell surface. But the minute we treat the complex with rapamycin to inhibit the mTORC1 complex, we begin to see the transporters process off the surface, be delivered to the lysosome, and begin to be degraded. And again, within 12 hours, they'll all be gone. So the ability to maintain transporters on the cell surface rather than come through the endosomal pathway is critically regulated by mTORC1. That bumped us in for the first time to the real central importance that others, particularly Michael Hall and David Sabatini, were talking about at the time about the mTOR complex and its critical role in integrating signal transduction, amino acids, and the energetics in response to stress to allow adaptive translation. And so again, rapamycin is the inhibitor of all of these processes, and downstream of this, TOR was considered the master regulator of cell adaptation to a variety of processes through its ability to control translation, longevity, cancer, diabetes, and metabolic syndrome, as shown here, but that did it through its ability to regulate and be a gain knob on the activity of translation of mRNAs into proteins. And so you can think about from their work that mTOR is a gain knob on the rate of translation so that cells can adapt or change or grow and proliferate. So we like that idea a lot, and we wanted to explore it further, how it is that amino acids influenced mTORC1. And so we turned a few years ago when we discovered macropinocytosis as an alternative strategy to ask, is mTORC1 downstream of amino acid uptake that comes from macropinocytosis? When you engulf proteins instead of free amino acids, take them to the lysosome, and then from the lysosome, you assimilate those amino acids. Our hypothesis would be that mTOR would be downstream in the model I just showed you, and that would then fuel adaptation or anabolic growth. So we tested that idea. And so we built some media that was deficient in a critical amino acid that cells absolutely have to have for growth. And that's the essential amino acid, leucine. And leucine is an essential amino acid. There is no way a cell can grow or proliferate in the absence of leucine, because leucine is the most common amino acid in cells. It makes up 7% of the protein mass of all cells that make up constituent cells. It's the most common amino acid, and it's essential. And so we wanted to ask and look specifically at the process of macropinocytosis in media that otherwise has no free leucine. So the only source of leucine is our addition of bovine serum albumin, a protein that could be taken up by macropinocytosis as a source for leucine. Now, as I've told you, as shown on the right-hand side of this slide, AKT transform cells take up their amino acids almost exclusively from amino acid transporters. And so what you can see, if you put them in media without leucine, you see that the cells, either the wild type cells or the AKT transform cells, are unable to grow. In fact, they begin to die and are almost all dead by 96 hours. And adding albumin does nothing to allow them to grow, although interesting, and we're still working on how, albumin's able to keep the cells alive to a certain extent in this situation. But that was in marked contrast to RAS transform cells. Remember, I told you RAS constitutively activates the rate of macropinocytosis. And so what you see here is that the wild type cells, again, are unable to proliferate in the absence of albumin, and RAS transform cells do no better. But if we now add to the medium, which is deficient in leucine, a 3% albumin as a source of leucine, now you see something fundamentally different. The wild type cells can live, but now the RAS transform cells can actually proliferate. And so that says that the leucine is being able to be captured and degraded sufficiently to be able to replace the need for leucine that is taken up by transporters in the cell. And that was exciting to us because for the first time we had an assay of when cells were using proteins as a source of amino acids to grow. And we wanted to show that downstream of that was TOR. That was our belief. And we couldn't have been more wrong when we did the experiment. We repeated the rapamycin experiment that I've told you earlier. We did it with an even better TOR inhibitor, but we've also done it with rapamycin. I'm just not showing this. If we take the same assay, culture cells in the absence of leucine in the media, and then ask them to proliferate, as I showed you earlier, if we add albumin to the media, we get the cells to survive. And now if we add taurin, a pan inhibitor of mTORC1 to completely inhibit its abilities, we see that in fact, when albumin is present, the cells now grow 5 to 10-fold faster, dependent on protein uptake and macropinocytosis as a source of leucine. And on the left is just a Western blot to show you that the downstream TOR1 substrates, that everyone that measures the ability of TOR to regulate translation studies, that is 4E binding protein 1, it has been completely dephosphorylated because TOR is inactive as a result of taurin, the T on that line, and S6 kinase is not phosphorylated. So TOR is completely inactive, and yet these cells are growing 5-fold faster than they were able to grow in the absence of taurin treatment. So that told us we didn't have the model exactly right. And we then wanted to make sure that, because chemicals, just for the graduate students and postdocs that are listening, aren't a particularly good way to interrogate a pathway, we wanted to do a genetic experiment. So we always also use shRNA to eliminate first the TORC1 and the TORC2 complex, and we got a profound effect when we eliminated the mTORC1 by knocking down Raptor. In the absence of leucine, cells deficient, the wild type cells and cells deficient in Raptor and Richter are unable to grow. That's what's shown in the upper panel on the right-hand side. That's what we've plated on a lawn, these fibroblasts that have been treated in these ways in leucine deficient medium, they can't grow after 3 days. In contrast, in the lower 3 panels, we've added albumin to the leucine deficient media. And what you can see is now the Raptor transform cells have done what normal fibroblasts. In 3 days, they've completely proliferated to spread out over the entire field. They have reached confluency. And as opposed to that, the control and Richter are not able to grow particularly well. And the growth curves are shown there. And you can see the rate of proliferation is dramatic and increased in the Raptor deficient. So both from genetic and chemical means, TOR is an inhibitor of the ability of cells to use macropinocytosis to fuel their amino acid needs. And we built an assay to figure out where TOR works in that pathway. So this is actually visualizing the uptake of albumin. It's done because we buy a commercially available BSA that's a fluorophore has been added to that's quenched into one of the binding poppers of BSA. So it only becomes fluorescent when the protein becomes degraded. So it allows us to look at and visualize the uptake into lysosomes and the degradation in the lysosomes of the BSA we've added. And what you can see is that wild type cells have a low basal ability when cultured in leucine deficient media on the upper panel, left to right, to take up into their lysosomes. That's the turning of the lysozyclic tract volumes, which are indicating in red where the lysosomes are at the start of the experiment. And they turn yellow as they degrade the green and dequench the green DQ-BSA. And so you can see there's a real uptake, and that's measured on the right in terms of its mean fluorescence over time. But if we add a TOR inhibitor, the rate goes up 20-fold. And the fact that TOR appears to be affecting the pathway is not in the rate of the formation of macropinosomes. The gain of knob on that is RAS, but on the rate at which those micropinosomes are fused to the lysosome, trafficked to and fused to the lysosome. And the dramatic augmentation is seen in the rapid rate at which all the lysosomes turn yellow in the TOR treated, and it's quantitated on the right-hand side. And so you can also do that with tumor samples directly from humans. These actually are mice, but we can also do it for human organoids. This is an experiment we did with Dave Tuveson's lab that's been published. If you treat pancreatic cancer organoids, which have RAS as their driver oncogene, and we treat them with taurin, and these are, you can see, starting in the internal cells, which are the most deprived of extracellular amino acid, taurin dramatically turns on the rate at which DQ-BSA is internalized and degraded in these cells, and it's quantitated on the rest side. So this works not only in normal fibroblasts, but in every cancer cell type and normal cell type that we've been able to examine to date. So that built a model, which is where we stood until the start of the new stuff I'm going to tell you about over the last 5 years, that said that yes, receptor tyrosine kinases and PI3 kinase initiate the signal transduction of a cell to take up substrates that will give them amino acids, either free amino acids or extracellular proteins, but that downstream, TORC1 has an essential job in telling which pathway to utilize as the source of amino acids for growth. Downstream, in an AKT-dependent fashion, the active TORC1 turns off the use of macropinocytosis as a source of amino acid and turns on and stabilizes the use of extracellular amino acids for take up. That makes a lot of teleologic sense. We're in the middle of the COVID epidemic, and the SARS-CoV-2 virus, including 60% of other viruses in its class, are taken up by macropinocytosis. Macropinocytosis is a risky strategy for feeding. It's a desperation strategy in multicellular organisms in the way we think about it, because you don't know what you're engulfing. So if you engulf a virus that's attached to one of the receptors that comes in through the endosome, as in the SARS-CoV-2, you internalize it, and in the lower pH of the endosome, as it's trafficked to the lysosome, it activates enzymes that allow it to penetrate into the cytoplasm. And that's the most common way viruses get into our body, including the SARS-CoV-2. So if you know that there's free amino acids as a cell outside, you'd much prefer to use those, because facilitated transporters are specific for the class of amino acids that are being taken up and can't be easily tricked to allow other or toxic agents to come in. And so it makes sense that you have a sensor to determine whether or not you have the simplest and safest substrate to eat to fuel protein translation, and mTORC1 appears to have that job. That idea fit a lot of ideas that had been developed in Boston at David Sabatini's lab, where he, downstream, along with Michael Hall in yeast, had come to realize that mTORC1 had a major role in sensing the intracellular amino acids that were available to a cell. And so we wanted to understand, how does TORC1, by sensing amino acids, regulate this decision to use extracellular amino acids or extracellular proteins? Um, and so at the time, the Sabatini lab, and this is a summary from 2015 when we started these experiments, had identified that intracellular amino acid concentrations influenced the TOR complex in terms of its activity. The first discovery was that mTORC1 is inactive when it's in the cytoplasm, and the active TORC1 complex that acts as a kinase has to be translocated to the membrane of the lysosome. So that said, it's likely to play a role there in regulating this trafficking part and sensing amino acids and determining whether or not you'll use the lysosome as a source or use the amino acids being taken up from transporters. That was very attractive to sorting out the two potential sources. David implicated a series of proteins in that regulation event that occur at the lysosome, including the V-type ATPase, specific amino acid transporters, the regulator complex, which I'm not going to talk about, the GTPases, RAG, A, B, C, and D. And it also was a site at which Reb negative regulation, which is controlled by growth factor signaling, is shown on the right, recruits and then allows TORC1 to be activated through the process of signal transduction. And what had been identified at the time we started these ideas is that there were two regulatory complexes that were required to regulate the activity of mTORC1 and its substrate preferences that existed to sense amino acids. They were the GATOR1 and GATOR2 complexes, and it had been discovered that amino acids activated, unfortunately, the negative regulator of negative regulation of TORC regulation, so that cestrin was a molecule that was identified that would inhibit the activation of mTORC1 at the lysosome, and amino acids would block the ability of cestrins to do that. We found that very attractive in regulating our model, and we went looking for how cells could sense that they're deficient in free amino acids that were being taken up and how that might be sensed by cestrins. We argued that this had to be a sensor that was independent of mTORC1 because it would control the mTORC complex. And so we started with making the media we just told you about. We knew that this was all regulated in leucine deficient media. So if we made media that was deficient in leucine, how would cells sense it? The most ancient pathway of sensing amino acid availability for translation was actually discovered in a strain of amino acid sensing of yeast years ago. And the first sensor was the molecule GCN2, the first component that's been figured out of what's called the general control network for recognizing nitrogen availability. And GCN2 is a kinase that is activated when it is bound by an uncharged tRNA of any type. So remember, the way you need to, why you need amino acids to translate is that you charge tRNAs with their cognate amino acid, and those tRNAs take to the ribosome amino acids that are allowed to decode an mRNA and then create an elongating polypeptide from the information in the mRNA as it passes through the ribosomal subunits. And so if an uncharged tRNA is present, that's going to stall translation, and the cell has to adapt. And the signaling mechanism for uncharged tRNA was discovered first in yeast, but exists in every metazoan organism, is recognized by uncharged tRNAs, recognized by the kinase GCN2. That phosphorylates a translational elongation factor, EIF2 alpha, to alter the properties of translation, slow it down, and alter the substrate transcripts that are preferentially translated, one of which is a transcription factor, ATF4. So we made media deficient in leucine, interrogated with a GCN2 pathway. We're able to implicate. I'm not going to show you the data. It was published in 2016, and found that, in fact, leucine tRNAs that are uncharged were capable of activating GCN2. That led to ATF4 dependent transcription, and that ATF4 transcription led to the transcription and translation of cestrins as part of the signaling. They were preferentially translated, and in the absence of leucine, cestrin 2 became the inhibitor of the mTORC1 complex through its ability to bind and regulate the GATOR complexes. And so that became a way cell could detect that there was an absence of leucine, free leucine, and it had to turn to proteins by turning down TOR activity and allowing then the fusion of macropinosomes to the lysosome to fuel amino acid uptake. That results was very satisfying with what at the same time David Sabatini was discovering about the sensing properties. Just as we discovered, leucine is sensed through the ability of leucine to bind to cestrin-2, alter its structure, and eliminate its inhibitory properties. So newly formed cestrin-2 will inhibit the Gator complex unless it binds leucine, and then it's eliminated. So that fit our data and David's fit perfectly. David was going on to show other regulators, particularly the Castor and mTOR inhibitors. Those two molecules sense directly arginine and methionine indirectly because the substrate of SAMTOR is S-adenosylmethionine, which is a directly converted to S-adenosylmethionine from methionine, so it's a sensor of methionine. And so very elegant description that TOR looks at key amino acids that are essential or conditionally essential, such as leucine, such as methionine, such as arginine. But it didn't really explain what would happen if other amino acids became limiting and how TOR would control that process. And so that became the new project that I want to tell you about in the last half of my talk. So we decided, because there is no condition that mammalian cells ever get cultured in leucine deficient media, except when idiots like people in laboratories make up media like that. There's no condition where you just lack leucine. There's not even a diet you can really make without incredible artificial synthesis that would be deficient in leucine, because leucine is the most common amino acid in all proteins. So we wanted to simply ask an experiment that had been used by the TOR field. What do you do if you just eliminate all amino acids? What will happen to these two sensing pathways over time? And this fit a notion, because TOR is a negative regulator of autophagy, of whether or not tRNA charging is ever a limiting factor for translation in real physiology. The TOR community did not believe that, but the GCN2 community believed that. And so we did an interesting experiment, which really informed everything else we did. Natasha did the following experiment. She cultured normal fibroblasts in the presence of complete media or in the absence of any exogenous amino acids, a standard experiment done in the TOR field. What you can see is in the presence of amino acids, S6 kinase is highly phosphorylated and GCN2 is not phosphorylated. So TOR is on, dictating increased levels of translation, and GCN2 is off, so it's not suppressing and altering the properties of translation. If you take away all amino acids, what you see in the second lane on the left-hand side is you see now TOR is completely off within 1 hour, and GCN2 has begun to be phosphorylated. If we add bafomycin to block the ability of macropinocytomes and autophagosomes to fuse with the lysosome to provide a source of amino acids to be degraded to resupply amino acids, either through autophagy or through macropinocytosis, nothing happens in the first hour, although you can see there's an accumulation of autophagic vesicles, which is the LC3 marker that's on the second lane when we add bafomycin. At 3 hours, we see that TOR is beginning to come back on as measured by S6 kinase phosphorylation, and it's even more strongly on at 6 hours. And if we inhibit autophagy and macropinocytosis with bafomycin, we see that the marker for stalled macropinocytosis of autophagy is on because LC3 is accumulated, and in fact, GCN2 is still accumulated under those conditions, and TOR has been repressed in its reactivation of S6 kinase. So that says the source of recovery of S6 kinase activity in amino acid-starved cells is really autophagy, the ability to eat your own proteins, degrade them in the lysosome, and resupply amino acids. And as long as that's ongoing, you can get enough leucine and arginine and methionine to reinitiate TOR-dependent translation. But interestingly, you still see under those conditions an increasing phosphorylation of GCN2, and GCN2 downstream of TOR is dictating altered properties of translation. And that really says that the cell still knows it's deficient in some amino acid that's being sensed by GCN2. And so the rest of this talk was, what's that amino acid? And what does that sensing tell us about amino acid availability? It's important to understand that TOR isn't a complete regulator of all translation. So on the right-hand side, what we show you here is the rate at which translation, bulk translation, is going on by just measuring incorporation of amino acids using an assay of new amino acid protein biosynthesis. And so we've looked at the translation rate, and at time 0, it's very high. As we culture cells over these same time periods, 1, 3, and 6 hours in no amino acids, translation rate doesn't go to 0. The best we could use as an index of 0 is cyclohexamide treatment, which poisons translation. And that's the rate when you use cyclohexamide, you could get incorporation in this assay into newly synthesized proteins. But what you can see is in the absence of free amino acids, as TOR is turned off, there's a profound decrease in total bulk translation, but it doesn't go to 0 even at 6 hours. You're getting altered properties because GCN2 is being around. I'm going to tell you a little bit about that as we go through the talk. So remember, tRNA charging is a quite complex and bioenergetic process, but the goal is that amino acyl tRNA synthetases bind amino acids, use ATP by converting it to AMP and pyrophosphate to charge the terminal end of the tRNA to its cognate amino acid as a substrate for translation. And that process is shown here. And we wanted to ask what percent of tRNAs are charged with their amino acid versus what percent of tRNAs in the cytosol are actually uncharged with their amino acid. Now, this was well worked out in the 50s, the chemistry of this, and there's thin layer chromatography that's extremely difficult to do reproducibly and to interpret, that was used in the 50s to study problems like this. But we decided this was a time ripe. Natasha took this on as a challenge. She thought it was a right time to figure out a next gen sequencing assay. This was the first step to her doing that. So on the left-hand side is what she did. She took classic chemistry that was worked out in the 50s to actually deprotect or destroy the 3' end of tRNAs that were uncharged by oxidation that would be protected if they were charged, and then do ligation of oligos to the 3' end and sequence those, as well as deprotect all amino acids and sequence them all. So she got the total tRNA pool and the number of tRNAs, and she could quantitate them that were charged. And so by that, we could measure tRNA charging. And she did the experiment over again, which is what's shown on the right side, of these key amino acids that we care about that TOR senses, and looked at that response. So on the right are the tRNA charging, fraction charged up to 100% for leucine, arginine, and methionine. And then, because glutamine is the most common non-essential amino acid, and one we had studied actively in the lab, we have glutamine, and as a control, we have valine, and I'll show you why we use valine specifically. What we were shocked to find is that at no time could we see any lack of tRNAs that are charged that would be needed for translation for any of the amino acids that TOR sense. There's still plenty of amino acids to charge those TNRs for leucine, arginine, methionine out the 1, 6 hours, and even 24 hours, which is not shown here. Valine, similar, another essential aminoid common in the hydrophobic region of globular proteins, was also not decharged. But what we saw starting when we took away the amino acids was the progressive and profound uncharging of glutamine tRNAs. And so glutamine turned out to be the most limiting amino acid for the ability to maintain tRNA charging. We wanted to make sure that that just wasn't an artifact of the assays that were built up because of the way we were building this up as a PCR assay. And so we actually used valine and incorporated, because there's a specific drug that was found in the 1950s that blocks valine tRNA charging. That's the drug Valinol. And so when you use Valinol, and do that treatment for 6 hours in complete media, you completely lose, or an amino acid deficient media, you completely lose valinol tRNA charging. But what you do is you recover all the glutamine tRNA charging, because now the most limiting amino acid is valine tRNA, because we've artificially done that with a chemical. So that says we could detect the recovery of glutamine tRNA charging in amino acid charged cells. We just had to poison another amino acid, and we could detect that in the assay. When we treated with valinol, we could see that the charge fraction of valine tRNAs dramatically went down. That's what's shown on the left-hand side. And then we were also able to see the uncharging of all of the essential amino acids if we added baphlomycin. As I told you, baphlomycin is necessary for autophagosomes to go to the lysosomes to recover their amino acids to continue to charge tRNAs for adaptive translation. And when we added baphlomycin, what we found is not just glutamine tRNA charging, but all the ones that were being maintained by autophagy went down, in this case, methionine and valine. And so that gets you to the most common when we first presented this in working groups around the institution, said that's all fine, but you just took away all the amino acids. And everybody knows in tissue culture, the most common amino acid that we supply in tissue culture is glutamine. It's 10-fold higher than any other amino acid. So it's not surprising that cells run out of it faster, because they have 10-fold higher. So we repeated the experiments. I'm not going to take you through all the other ways to convince ourselves that this glutamine is truly specific. But one simple way to do it is to just take and dilute all the amino acids progressively together from what they were in medium. So we start with glutamine 10-fold higher than any other amino acid, and then we just reduce the levels of all amino acids down to 5% of what they exist in common tissue culture media. And again, when you look at tRNA charging in the same spectrums as we do, we find that after 24 hours, glutamine tRNAs are completely uncharged, and GCN2 is fully activated, and no other amino acid transporter that we could find was charged. But we wanted to absolutely convince ourselves that that was right. And so Natasha took this validation to build out the full next-gen assay that I want to show you very quickly. We wanted to do that because at the same time we were doing these experiments, we were measuring amino acids in human tumors that were isolated. This is an experiment we did with the head of surgery here, Jeff Drebin, as well as Josh Rabinovich at Princeton University. And much to our surprise, we found in naturally occurring tumors, as they outstrip their vascular supply ability to bring in oxygen and amino acids and glucose and pH, that the most profound amino acids that were depleted in the tumor environment were in fact the essential amino acids, in which glutamine was consistently the lowest depleted. So this again focused our attention on glutamine and said we really want to be absolutely sure that glutamine tRNA charging is specifically compromised. And if you really paid attention to the earlier assay, we were just looking at one specific codon's tRNAs. We wanted to look at all of them. So Natasha built out an assay to measure all tRNAs. And then she used a trick, which we had discovered from tumor biology 10 years ago, which was that in growing cells, both tumor cells as well as normal cells like hematopoietic cells or immune cells, the primary use of glutamine is not actually for tRNA charging. It's actually used to make other amino acids, nucleotides, but the most common use, as we reported 10 years ago with Ralph D. Baradinas when he was in the laboratory, the most common use of glutamine was to capture it by deaminating it through glutaminase to glutamate, to both supply glutamate as a source of building other non-essential amino acids, and also being deaminated glutamate in the TCA cycle to provide TCA cycle anaporosis. And that when we measured it, 90% of glutamine in tumor cells was used for that process. And that and growing cells used it for that process. That suggested that maybe the reason why glutamine is so consistently depleted in amino acid environments is because of this use for a process other than translation through the processes of glutaminase. So Natasha built out an assay. I guess I put this in a different order. I apologize. She first asked if that was real, that was really the cause of the depletion of glutamine. And so she got a drug that is now available to all of us. It was an inhibitor of glutaminase that's very specific. And she asked, could she recover tRNA charging by inhibiting glutaminase to prevent the conversion of glutamine taken up into the cytosol to being degraded to glutamate in the mitochondria? And when she did that, she got a dramatic and highly statistical and highly reproducible recovery in translation and glutamine tRNA. So this is actually just using glutaminase to recover the rate of translation in amino acid deficient media. So with that, she built out the whole assay. That assay now depends on the same chemistry at the front end, making cDNAs from the oligo that's primed, but then to develop ligated adapters that are specific for every single one of the tRNAs that have cognate amino acid receptivity, and to do that with barcodes so that we can identify them specifically and their charged and uncharged. We can recognize at any one time in any cell type that we've looked at about 185 of the tRNAs of the about 225 are coded. We don't know whether that's because we can't see 50 or that in fact the cells rarely make more than 185 meaningful numbers of transcripts, more than 185 of their tRNAs. We can see every single coding codon in her assay, and I'm going to show you how we present that data right now. Here are all of the amino acids and all of their cognate tRNA recognition codons. And what you can see on the left-hand side is we grow cells in complete media with optimum growth conditions. Every single one of them is charged. That's why they're in green. If we take away all amino acids for 6 hours, as I've told you, we get very selective uncharging of one set of codons, and they are all the codons for glutamine. No other amino acid goes down. And then if we add glutaminase, what we discovered in this cell type was that we can recover glutamine tRNA charging to a baseline level, but now methionine becomes the limiting amino acid. I'm not going to tell you anything because we found different answers for what becomes limiting when you recover glutamine in different cell types and different cellular conditions. And that's something she's going to be working on in her own independent lab when she works on, and I'm not going to talk about that. I am going to talk about why in every cell and every cell line that we've measured, glutamine always reads out as the selective uncharged tRNA in this assay. And this is just the quantitation of that's just on the right hand of this slide. But this shows you that we can measure every single one of the codons and their tRNAs. So we wanted to make sure it was specific. And so this is a slide that's a composite that I got. I just want to show you the right-hand slide. The only amino acid that we can add to amino acid deficient media that will recover translation is glutamine. Even methionine does not recover it, even though it's the most common secondary amino acid if we, as I just showed you, if we do glutamine in cell lines that we study. So glutamine is truly special in this assay, and it is truly the thing that GCN2 and amino acid deficient environments of all kinds is recognizing to drive its properties. It's a glutamine sensor, GCN2, in our tissues. One surprising factor that we found that drove the rest of our work was that in addition to translation going down, we noticed that transcription was profoundly impacted when we put cells into amino acid deficient media. So that's what was shown in the first samples. This is the rate of transcription, or the rate at which we incorporate a measurable 5-ethinyl uridine into messenger RNA in baseline, and what happens after a few hours in no amino acid containing media. That we were surprised to discover, which made us interested in this, was also that decline in transcription, which you just said cells can tell they're just not having resources to translate, and ultimately this is just a secondary consequence. The surprise came when Natasha treated those cells with glutaminase to recover glutamine tRNA charging. Immediately transcription went back up. And we could do exactly the same thing by supplementing amino acid-free media with glutamine, as shown here. And so this was glutamine itself was being sensed by bulk transcription, and we became interested in what did that mean about the proteome as it changed when there was no translation or limiting translation because of glutamine tRNA charging had gone down. And we got a dramatic answer that the proteins most specifically affected were of one specific class. When we did proteomics, the proteins that disappeared when in low amino acid media or when cells were cultured with no amino acids all had one common feature. They had polyglutamine tracks. The two big classes of proteins that have polyglutamine tracks are components of transcriptional initiation, Pol2 regulation, such as the mediator complex, TATA binding protein, as shown here, and then genes associated with neurodegeneration, like Huntington's disease, and other proteins associated with neurodegeneration syndromes. All of those proteins, when we measured them, they're called polyQ containing proteins, specifically disappear when we culture cells in prolonged periods of time with no amino acids or 5% amino acid cocktail mixes. That's shown here on the left-hand side on a Western blot. And we can recover specifically the levels of polyQ protein by adding either glutaminase inhibitors or by adding specifically physiologic levels of glutamine on top of the rest of the amino acids being deficient. And that's quantitated on the right-hand side here. So this said that tRNA charging of glutamine amino acids was being specifically sensed by the decrease specifically in the translation of polyglutamine containing proteins. And so glutamine tRNA change was being sensed because a test was being run on these proteins to reiteratively add glutamine to the elongating polypeptide. And when 95% of the tRNAs are uncharged, that fails and the nascent polypeptide falls off the ribosome and is degraded. And you can only recover that by recovering enough glutamine to recharge tRNAs. We wanted to make sure that was specific for glutamine, so we cultured in cells not just in low glutamine media, but in comparable limitations of leucine, because I've told you how important leucine is to translation, so that global protein translation is affected by leucine, and asked, does that, when we inhibit, because there's no leucine tRNA charging, would we not see the decline in polyglutamine proteins? And what you see on the right-hand side, the left-hand side is the control. It says at low glutamine, which is measured at 300 to 400 micromolar, or low leucine, about a 7-fold less leucine, 60 micromolar, you get a comparable amount of repression as measured by puromycin incorporation in nascent proteins of translation. However, when you look at the proteins as they're being affected, what you see is that there's the low glutamine condition, there's specific decreases in the polyQ proteins, including the mediator complex, TATA binding protein, CBPF and CBP, but you do not see that change in leucine deficient media. Those proteins are fully retained. So this decline in polyglutamine is specific for the limitation of glutamine tRNA charging. And beyond that, this works, this sensing by regulating the levels of mediator and TATA binding proteins is regulated in the physiologic range in which glutamine occurs. In extracellular fluid, in viable tissues, the range of glutamine is between 0.3 millimolar and 0.5 millimolar. And you can see as we add to 5% amino acid progressively back glutamine, the level at which we begin to regulate dynamically the level of things like mediator 12 or CBPF alpha or TBP occur exactly in that range. And so this is built to be a sensor of the availability of glutamine in cells. To really get a sense of whether this happens in vitro, Natasha built a reporter assay in which we built an exon, a naturally occurring exon from the Huntington disease gene, which has 36 polyQ repeats, to a bridge peptide, to a GFP reporter. And to make this not work, we put it one base pair out of frame because it was known when When ribosomes try to go through iterative and get stalled, they'll often back up or advance one base pair and mistranslate when there isn't sufficient charged tRNAs to drive them forward to see if, in fact, so the in-frame codon would incorporate polyglutamine, and that would put GFP out of frame and it would not read. But if it backs up one nucleotide and then reads through, you'll get a polyalanine track and you'll translate GFP. And so Natasha was able, so here's just a pancreatic cell line, but we've done this in multiple cell lines. If we transfect in this reporter, we grow that in complete media, or we treat with glutaminase complete media, there's no change or no baseline GFP in this assay at all. We grow cells in low amino acid conditioned media, and now the reporter turns on in virtually every cell because you're mistranslating because you can't get through polyglutamine tracks. And if we add glutaminase inhibitor, that collapses back to the menu. As we recharge by inhibiting glutaminase, we reach glutamine tRNA charging. And so we have taken that assay forward. The lower part of the panel is just some additional controls, particularly on the lower left-hand side. What we've showed you is that this GFP translation isn't just a response to stress in general. The only stress that will eliminate it is amino acid limitation. When we limit glucose, it doesn't turn on. We do a stress test with hydrogen peroxide to go oxidation stress, it doesn't turn on. Calcium stress does not turn it on, and salt stress did not turn it on in this particular experiment. So this is a very selective activator of the polyQ track, and it requires the polyQ track. To do that, we did that by making a, so again, this is just reiterates that. I'm sorry, I didn't realize it was on both panels, but again, this is another experiment where we tried stressors. The only stress we can find that turns off is amino acid stress. And it's very important that this occur in the naturally complete proteins. So we built TBP with an HA tag so we can detect it and transfect it into cells. And then we made TBP without its polyglutamine track. So it's the same protein. It just doesn't have a polyglutamine track, which is not required for TBP function, as others have shown. And what we can say, it requires the polyglutamine track to be regulated by low amino acid levels. So when the polyglutamine tract is level and you eliminate amino acids, it goes down. We add glutaminase, it goes up, and there's no effect on the HA thing when we, in fact, or even the opposite effect, when we eliminate amino acids and there's no polyglutamine track. So this appears to be specific for proteins with polyglutamine tracks. And then finally, to demonstrate it's as a proof of principle, we made tumor xenografts and we interrogated whether or not this reporter would read out. This is an assay where the assay that's being read out is recognizing the GFP with an antibody. So it's the brown regions. And what you can see in hypovascular regions of this growing tumor, we see nests of cells that are experiencing a profound alteration in their ability to translate through polyglutamine tracks. So that's where we are. We're building out this assay now to look in a wide variety of situations. Natasha's looking in neurodegenerative diseases to see if we can use these kinds of other reporters that she's built out on these principles to interrogate and to be interrogating in vivo conditions for when glutamine becomes limiting. And so that's where we are. I'm pleased to have had a chance to talk to you. I do want to say the one follow-up experiment we've done to the in vivo experiment is to isolate cells like this and convince ourselves that they could recover, that they're not N-terminal cells that will die, but can actually recover. And so we have done that, which is this last experiment. But since I'm going to the end of time, I apologize. This is my first time of presenting any of this data in public, but it allows us to validate the importance of TORC1 and GCN2 as regulators of translation and their critical role in disease and adaptation to real natural environments. And with that, I'm happy to stop and take any questions. And I will just let you, for those who want to hear more about this, Natasha will be giving a talk at Harvard Public Health School in the next few weeks, and she'll be expanding on some of the things I talked about.
Terrific talk, really stimulating for many reasons. We've got really a number of questions. We're already over time.
I apologize. I really
No worries, no worries at all. Maybe I'll ask one that I can't resist. Do you think the gain of the polyalanine tracks could be adaptive in some sort of neomorphic way?
So we've talked a lot about that. There's many things that we think are logical questions downstream of this work. That is one of them. We also are very interested in polyasparagine tracks, who as you know, in Boston, and when Sue Loon class was working on this in yeast, gave a different information of stress response as opposed to polyglutamine tracks. So we think we want to integrate it into those kinds of studies, but we think repetitive amino acids are sensors. And this work proves glutamine is, but we're pretty sure arginine, or asparagine is as well, as is other like polyalanine tracks. So this is I wish I knew the answer, but this is where we are. We're pretty comfortable every way we can test it. Glutamine is, we're getting comfortable with asparagine, but we have not yet tested alanine, but we are fully aware, given that that's how the reporter works, that that's likely.
Great. Well, unfortunately, we're out of time, but I will, if you don't mind, we'll email you the questions so you have some time to ponder, and we'll hopefully be able to
I greatly appreciate any of the questions. How we get better as scientists is by this exchange. And so once again, I apologize to the community for not a broader discussion of this. I actually thought, since I only had 37 slides, I'd be able to do it, but this is my problem of not giving enough talks recently. And so I took too long. I apologize.
No problem at all. Pleasure having you. Thank you, everybody.
Thank you all. Take care.